Okay, so you can see in front of you the program for this lecture series that's going to run every Tuesday and Thursday through to the end of March. And we have a series of talks on various biophysical techniques that are all available within the LMB, obviously available to LMB researchers. But we like to try and promote biophysical techniques as an approach to studying structural biology. And so we hope that the information is useful to people outside of the LMB. The talks are being recorded and you'll be able to access the recordings eventually on the external LMB website if you wish to hear the talk again. So what we're going to do in terms of questions to the speaker is if you could type your questions into the Q&A box. You should be able to access the Q&A box at the bottom of your Zoom screen. And during the talk or at the end of the talk, if you would like to type in your question, Nick will read through and work through the questions and provide answers, hopefully, at the end of the talk.
Thank you.
So without any further ado, I will hand over to Nick and we'll hear about light microscopy. Can you share screen?
Yeah, I'm just getting there.
Okay.
Where are we? Here we go. Share. All right. Okay. So I'm just going to give a quick overview of light microscopy and confocal microscopy. There's quite a big subject area, so this has been split up into a number of lectures. Anyway, I should give the starting one. So, there we go. So the facility actually at the LMB is got quite a few members of staff, but we're not all doing exactly the same thing. So it sort of splits up into three areas. There's me, Nick Barry, and then John Howe and Ben Sutcliffe sort of deal with the day-to-day actual running of all the instruments and looking after them. Jerome Boulanger is our expert in image processing and image analysis. So he can basically come up with some he'll sort of help that side of things, the downstream side of things. And then James Manton is actually pretty much spends his time doing instrument development, so there's sort of a separate section. But if you use any of the light sheets, you might come across him as well. There's a lot of machines in the LMB. They are sort of pretty much high-end confocal microscopes, turf microscopes, super resolution microscopes, stuff like that, the sort of instruments that typically end up in facility. I will just highlight, we do have a light sheet microscope. So this is a picture of an older one, actually. That's got its own separate talk, because that's quite an important area. So I'm not really going to cover that one at all today. I'll cover sort of microscopy, fluorescence, confocal, and then end up in multiphoton microscopy. But we are in different times now. So it is light microscopy in the time of COVID. What does that really mean for working at the moment? Is basically reduced room occupancy. So John and I and Ben, we just can't really go into the same room and sit down with you anymore and use the microscopes as we used to in the past. You'll see that there's sort of hygiene kits and a wipe down protocol so that you don't pick up contamination from the microscopes themselves. The training is now done by remote access to the microscope computer and by a Zoom call on a separate device. So we're still able to train you. And plus, there's a lot more remote logging into analysis computers and even into the microscopes themselves. But what I do want to emphasize for those at the LMB is that the staff are still available for help and troubleshooting. So you can just email us, we can Zoom in to you while you're working on the microscope, and we can remote log in. So we can still help you use these instruments effectively. The sort of things we're looking at vary quite a lot. From here is some bacteria. So here we're really looking right down at the diffraction limit. This is such skeletal structure within a number of bacteria, 1, 2, 3 bacteria here. bread and butter work is obviously sort of images of cells grown on cover slips, in the nuclei, the cytoplasm, and so forth. But it goes up to sort of model organisms. This is a Drosophila embryo, and you can see it's sort of internal organs have actually developed by this point. That's done on a light sheet. And then going up to tissue slices and things like that. So this is a bit of a mouse brain. So that's of the order of a centimetre. This is something that one could zoom into and see quite a lot more fine detail than is evident here. So the other strand of thing that I alluded to earlier on was this idea of quantitative imaging. So it's not just collecting data, we want to do something with it, and that's about image analysis and so forth. And that really is the area that Jerome is our expert in. But oftentimes that starts with just segmentation of images. Here you see cells separated out into individual cells. So we have software that allows you to do this in quite an intuitive and step-by-step manner. This is not sort of stuff that isn't a black box, but also equally, this is scalable. You can scale this up to sort of thousands of images on plates and what have you. So one useful question that we asked just right at the sort of basics of microscopy is, when was this really invented? Well, lenses do go back a long way. We can sort of, the optics of, serious optics in terms of sort of scientific and what have you, in the sort of, certainly in the European framework, is often sort of benchmarked against the work of Galileo with his telescope. But even before that, people like Zacharias Janssen was claiming to have invented a microscope, where we don't have a great deal of knowledge about what he actually did with it. The really important ones, I think, were sort of in the sort of middle to late 17th century with Hooke and Leeuwenhoek in the sort of 1665, 1670s era. They were actually doing what we would recognize as biological microscopy, and the sort of general principle of the microscope is indicated by the sketch below. You can see a sort of little object slightly a distance away from a lens forming a much bigger image. So this is the general principle of the microscope, but as we see, it's a lot more than that. With Hooke, I mean, this was really a landmark. This was a person looking into a world that no one was aware of. This really was the advent of microscopy as we would understand it in the biological context. And his output was drawings. He has a big book, Micrographica, full of these beautiful sketches he did. And he was the person who described the cell. But what is interesting to know with sort of Hooke and his contemporaries is they were looking at a world that they had no idea existed. I mean, here we know about bacteria and things like this and what have you, but in those days there was no inkling of these things, and they were able to faithfully reproduce pictures that stand the test of time just by observation and drawing. The other big figure, of course, is Leeuwenhoek, who published his works a little bit later, but basically these were contemporaries. Here's a picture of his actual microscope. You can see this little black dot is actually a simple lens. It's a very short, nearly spherical lens mounted in this sort of brass plate. The sample was stuck on this spike, and then the rest of this mechanism here is all about moving it around and focusing it. And you just looked through from the backside with sunlight coming in behind the specimen on that spike. So again, I just alluded to the principle of the microscope. The object is slightly further away from F than the focal length of the lens, and produces a magnified image over here. If I turn that around, I mean, that does look much like a sort of cell phone camera. You've got, people standing in a group several meters away from the lens, from your cell phone, and it produces an image on a sensor which is built in within the thickness of the cell phone. But if you take that analogy, you realize that microscopes are not meters in dimension. You don't stick the sample in within arm's reach. And likewise, cell phones, you don't generally see the fine detail. You can't see the sort of individual stitches on this jumper of somebody who's standing in front of it, what have you. So what is special about microscopes? They really are optimized to reveal the fine details. So you need two things for that: magnification and resolution. And in this case, we're actually trying to look at things that are smaller than the wavelength of light we're working with. So visible light, sort of 500 to 700 nanometers. We might be trying to look at a vesicle of a few hundred nanometers in dimension inside a cell. So this is slightly different to telescopes, which are optimized basically to look at very, very distant objects. But so this means that microscopes have some sort of difference in their sort of conceptual differences in how they're made. Just to go in terms of biological samples, we can work sort of all the way from animal models down to sort of bits of tissue, fixed tissue slices, single cells grown in culture, subcellular organelles within them. And then at some point we sort of basically run out of the ability to visualize stuff. And this is certainly well before single molecules. So microscopes do have this optical resolution limit at the bottom end. And equally on the other direction, as you go to bigger and bigger samples, you've got ever increasing scattering absorption, which degrades the image. And that's dealt with slightly differently. So the first goal was really to get some sort of resolving power. And you've got to deal with these sort of three different aspects of light in optical instruments. That's diffraction, refraction, and dispersion. So diffraction is about the wave-like nature of the light. Refraction is basically just the optics of building lenses and what have you, and how you deal with the aberrations that are intrinsic in spherical surfaces. And then dispersion is the sort of wavelength dependence of optical devices. Obviously, the key thing though is that there is a limit, and this limit is set by diffraction. And basically what happens is when you want to look at an object using a ruler, effectively the wavelength of light, that when they're to a comparable size, there's got to be some sort of consequence of that. So the sort of modern version of this sort of idea is, you take some plane wave, put it through a small hole, and form an image of the diffraction pattern at the back focal plane of a lens, and you get a pattern like this. So this is diffraction in sort of, as we would understand it. Now, obviously, this is easily done if you've got a laser and what have you. You get these really brilliant and bright diffraction patterns. But in the sort of 17th century, there obviously were no lasers, so it wasn't quite as straightforward as we might think about it. Also, there was a lot of dispute about what the true nature of light was. So, things like Snell's law are, really were sorted out really at the dawn of optical instrumentation. And actually, the Ptolemy and the ancient Greeks even had a measurement of bending at surfaces due to refraction. But there was the whole field was sort of muddied by this sort of camps saying that light was a particle and camps saying that light was a wave. And Huygens came up with this idea towards the end of the 17th century of secondary wavelets, was actually got very close to what we would how we would treat modern optical instruments. Newton was largely wedded to the idea of particles, and his voice was so strong that it would drown out others. But the really important experiment was done by Thomas Young right at the beginning of the 19th century, and this was his Young slit experiment, which demonstrated interference, and that really sort of said it was wave-like patterns, and you could see these diffraction patterns. So he was able to show these diffraction patterns right at the turn of the 19th century using a very narrow aperture and illuminated by sunlight. But this wasn't enough to really work out how optical instruments work in terms of getting them to resolve fine details. This was a problem that Zeiss, for example, had come up against when building microscopes, and he got this guy Abbe to basically help him figure out what was going on. And between them, and particularly with Abbe, he came up with a theory of sort of image formation in optical instruments that really has stood the test of time and describes why properties of the microscopes have to be as they are. what Abbe did is had a pretty simple experimental apparatus, but what he did was to reduce his sample to a sort of a test object, a very simple test object of just diffraction patterns. So basically lines on a slide, and he put various filter stops inside his microscope, these little patterns that let bits of light through. So he basically was aperturing the back of the lens or aperturing the image plane. And with this, he was able to figure out what was important. So the idea here with this sort of diffraction pattern type object is that the finer the pattern, the more the diffraction orders are pushed out. So here's a coarse grating with big gaps between the lines. You can see the primary zero order light getting through in the back focal plane. But you can also see there's a first order diffraction pattern. You can see there's some dispersion depending on wavelengths from blue to red. And then the second order is just in the sort of field of view of this box here. If you go to a finer pattern, you can see that the first order gets pushed out to a bigger angle and the second order has disappeared on this sort of box idea. So this concept of diffraction orders is key to the work of Abbe. The other thing that Abbe did was come up with this definition of numerical aperture, which really helped simplify the sort of description of this problem. But one thing that it shows us is the refractive index of the sample is really important in NA. So oil, which has a refractive index of 1.5, is going to give you a higher NA than air, which has a refractive index of 1. So here NA is king in terms of resolution. So this is sort of Abbe's experiment in a sort of sketch type form. We got this sort of hypothetical object of sort of perfect contrast, light, dark light, dark light, dark light, that's producing diffraction orders. And in this sketch, you can see that some of these diffraction orders make it into the collection cone of the lens and some don't. And Abbe's formula in the end, which he came up with, was the resolution of the microscope is related to basically 1.22 times the wavelengths you're working with divided by 2NA. So this is really cements the idea that this aperture, the collection aperture of the lens, is really important for resolution. Incidentally, I mean, obviously a higher NA gives you more light collection, so a brighter image. So Abbe's reasoning goes something like this. You have this object on the microscope stage. It's got some fine structure, which leads to diffraction. The lens only captures a limited number of these diffraction orders. So basically, all the detail of the object is not reproduced. If you've only got one order, you'll get a sinusoidal pattern rather than a square wave pattern. And the more orders you get, the sharper these objects, these transition from black to white become. So that's sort of the sort of basics of the thing you cannot escape from due to wavelengths of light and diffraction. The other thing is refraction, how lenses bend rays of light. So in reality, because of lenses are spherical surfaces, not all the rays end up in this sort of perfect diffraction limited focal spot. So there is some residual aberration, and modern optical engineering has really got rid of a lot of those. But we as users actually do introduce aberrations ourselves, which can distort the performance of the microscope. So I'll just go into that a bit more. So if you consider this simple case here, we've got these three rays converging to a point. So this is sort of a diffraction limited spot, let's say. And if we got a high quality plan apochromatic lens, which often is the case on the confocal microscopes, and we use them exactly as the lens designer intended them to, these images would be almost free of aberrations. However, if we stick a slab of refractive index material into this light path, you can see that because of refraction at these surfaces, these rays now cross at different points. So this ray here and this ray here are now crossing somewhere over here, whereas this ray here and this ray here are now crossing down here. So this is this sort of the basics of aberration, because of refraction at every surface, every interface of refractive index. There's also the fact that these angles here are wavelength dependent, which leads to chromatic aberration. I'll get to that in a moment. Okay, so in many respects, one can design the objective with these properties in mind, but the problem is the sample isn't that necessarily that straightforward. If we think about an objective here up at the top of the image, we've got the sort of last element of the objective, a lens with its own refractive index, some immersion medium, a cover slip, and then the sample. So we've got these potential different refractive indexes, and they all look conceptually like sort of slabs of material. So this idea of rays ending up in the wrong place, you can see is going to be built into the microscope when we put the sample on. Immersion medium could be air or water or oil or maybe some other sort of fancy medium, but they're all effectively like slabs. And what's happening is, if as you go through these parts to get to your sample, their effect becomes, it impacts how good the microscope behaves, particularly if you use a cover glass that's too thick compared to what was built into the design of the objectives. So really, we want to use a number 1.5 most of the time, which has got a thickness of 0.17 millimeters. It does matter if you use one that's 0.2 millimeters thick or 0.1 millimeters thick. This will have an effect. If we mount the medium with a lot of gap between the sample and the cover glass, and this mounting medium is not refractive index matched, then we'll get an effect at this interface as well, and so on. So the sample does have an impact on what we do. So it's really worthwhile taking care of these details, and so we can help you get that right if you want to, if you ask us. We have mounting media and we have cover glass, and obviously we prefer that we provide the right immersion media for each microscope as well. So as a rule of thumb, use Zeiss immersion media on a Zeiss microscope, Leica immersion media on a Leica microscope, and so on. So the last thing I wanted to talk about on this aspect is dispersion, the spreading out of the spectrum of light by a prism, for example. So this was actually sort of solved towards the end of the middle of the 18th century. I think I can't see the numbers up the top there, and with the achromatic doublet. This is actually a sort of a clever combination of one type of glass that has got a lower refractive index and another type of glass with a higher refractive index, and the two together formed into this sort of compound lens of two joined components, conspires to actually send sort of the red and the green light back to the same point. So with that, you lost this sort of rainbow of halo around objects that was common in prior to this. So this was sort of around 1830. You got sort of a microscope that was chromatic corrected and spherical aberration free. This form of microscope is less and less used these days. The modern one is somewhat more like this. So the sort of older style, you have an object slightly more than f away from the objective, forming a real image somewhere down where the eyepiece is placed. So the object plane and the image plane are sort of said to be conjugate to each other. Modern microscopes have this format where basically the object is placed f away from the objective, so that the light from a point comes out as a collimated ray bundle. And then you have something called the tube lens, which actually then forms the final image at either the eyepiece plane or the camera plane, for example. What this allows you to have is this sort of gap here between the objective and the tube lens that you're sort of free to put things in. You're also free to vary this distance. So this is useful if you want to build a microscope platform that has sort of lots of different modes. It could be a white light microscope. You could insert a whole bunch of optics in here for doing fluorescence microscopy. You can bring in things for doing manipulating the sample, like sort of a photo bleaching beam or something like that. So in this gap, this so-called infinity space, is where you've got the freedom to do that, and you don't really have that here because this distance from object to image is rigidly constrained, whereas only really the distance from the tube lens to the image is rigidly constrained. This bit, you have some flexibility. Again, modern objectives are covered in writing. You've got a manufacturer, the sort of type of objective it is, magnification, what its NA is, what its immersion fluid is, whether it's an infinity correction thing and cover slip. So looking at all of this can get you, can tell you basically how to use the objective. And generally speaking, the more writing you have, the more it costs up here. So taking a look at that is quite informative. It can help you put your sample on correctly, particularly it'll tell you the cover slip thickness. And there's usually a color coding on them, which indicates what their magnification is. So types of objectives we typically use, we have the sort of lower magnification ones, normally air, so they've got a big air gap between the sample and the objective. This is useful if you're looking at things in plates or things that you're looking through thicker plastic things. You've got enough working distance there to actually go through to work with a high-sided plate, for example. Once you go to higher magnification, typically they're very, very close to the sample, and there's some refractive index matching thing in there to get the NA up. So with an oil immersion, we might use a 1.5, 515, 518 oil immersion medium, which means that the NA can be a number higher than 1, and that gives us higher resolution. So if you're using oil, you've got to be wary of refractive index mismatch, because normally what that is, the assumption is the sample itself is mounted in something that's almost oil-like in refractive index as well. Intermediately, there are these sort of higher magnification air objectives. These are very useful for sort of doing screening work where you're moving from sample to sample to sample, but you just want to make sure that they've got the reach to get through the mounting of your sample, so they don't necessarily work through thick plastic bottom plates. They will work through a cover slip. For very high NA, you've really got to make sure that cover slip is just the right thickness for it to work well. For live samples, we can switch into water immersion because we have our sample in a sort of watery environment, a water immersion medium, and then the objective. So really, this sort of scenario is one that allows you to focus up and down, because effectively you're just swapping water on one side of the cover slip and water on the other when you focus, and the optical properties are maintained. And then the final sort of class of these sort of so-called dipping objectives, where you have basically a longer gap between the sample and the lens, with maybe 2 or even up to 3 millimeter column of water in this gap here. There's no cover slip, so these are good for EFIS and what have you, because you can stick an electrode down in that gap there. So normally this angle on the objective is much more sort of conical rather than flat. What about looking through the eyepieces? And obviously we're not looking at an image that's directly at an image that's coming out of the microscope. We put the eyeball right up close to it, and you can see where my hand is now. It's, we're doing this. I can't really see my hand, but the eyepiece in itself is a little optical instrument that puts the image a comfortable distance away from my face so that I can actually look at it. One thing to notice is you'll see this sort of this graduated setting on many of the microscope eyepieces. This is the so-called diopter setting, and it's equivalent to your glasses prescription if you have them. So if in nothing else, just make sure that you set that to 0. And also, if you've got two eyepieces, make sure they're set the same, so you don't have your sort of one eye trying to compensate for something here and the other eye trying to focus there, because that's a sort of bit strenuous for your eyeballs to do focus the different length distances. The simple eyepiece was early. It was an early invention. This is effectively by adding an eyepiece, you've got a two-lens microscope or a compound microscope, and what it does is that the lens is actually closer than f away from the real image formed by the objective. I've been somebody up in the interstitial bashing on a pipe. It's not very good for the acoustics here, but I'll battle on. What this does is it forms a virtual image. So rather than having a real image, a smaller real image here, the eyeball sees a virtual image, which is actually a sort of a comfortable distance away, and it's got some extra magnification. So when you're looking through the eyepiece and sort of ask yourself the question, where is that image actually? Where do I think the image is? You'll sort of think, oh, well, it's something like 30 centimeters, half a meter away from me, in the sort of in virtual space inside the microscope. But if we look at all the components of a typical microscope together, you realize that there's a considerable complexity in them. We have some sort of white light source, let's say, a condenser optics for illuminating the sample, the actual specimen itself on a slide, and then the objective and an eyepiece, and then this lens here represents the lens of your eyeball, and ideally forming an image on the back of your eye. So what we have in these microscopes is this concept of conjugate planes. So if we just flip over to this one first, we have the real specimen, an object, and it forms an image on the back of your retina. So we have this image here is conjugate to this one, and then intermediate just before the eye, somewhere close to the eyepiece, there's the intermediate image. So this is conjugate to that as well. This would be where you'd put a camera if you had a camera on the microscope, but obviously with an eyepiece, we've got some more optics, including the lens of the eyeball, rather than in this instance the camera, but that forms a real image on the back of your eyeball. So these are all conjugate planes. So there's another set of conjugates to do with the illumination source. So if we think about this as a sort of the filament of a tungsten lamp, what we definitely don't want to do is put a filament of a tungsten lamp on the sample. We want sort of uniform illumination. But alternatively, to get lots of light through the system, we want the sort of image of the tungsten lamp to be sort of fit conveniently inside the rear aperture of the objective, so that basically all the light from here can exit the objective here. So this is the sort of, these again are conjugate planes. This filament is conjugate with a rear objective rear focal plane. And then when you put your eye up to the microscope, there's a sort of an eye point. There's a sort of imaginary aperture in space that is the entrance aperture of your eyeball. So we basically want to match the hole in your eye, the pupil of your eye, to the exit pupil of the eyepiece. So again, these are sort of conjugate planes, and if we match those pupils, then we're going to get efficient transfer of light from the illumination source into the eyeball. But unfortunately, it tends to be the case that you really got to, if you want to get the best out of your microscope, you've got to actually focus that condenser. If you go to a shared microscope, the default condition is that the condenser is going to be out of focus, out of alignment. So taking care of that will actually improve the image quality, because you basically get the most uniform illumination of the sample, efficient use of light. It does impact resolution. You minimize background glare and scatter, and it sets your microscope up for contrast enhancement mechanisms such as phase contrast or differential interference contrast. So overall, taking care of the condenser gets you the best images. So if we just quickly go back again, focusing this set of optics correctly relative to the sample and to the objective gets you a higher resolution image. So typically we use something called Kohler illumination, and what that requires you to do is to identify some of the parts of the condenser optics, so the condenser focusing knob and the centering knob. And then there's usually some iris diaphragms, a condenser aperture and a field aperture, which you need to adjust as well. But first of all, you've got to put a sample on the microscope and focus it. Then if you close down the field aperture and focus that on the condenser, that effectively is the step that gets you a focused condenser. You'll see a sharp image of the aperture of the condenser in the image that you're looking through the eyepiece at. Then you can center it. Then if you just open it so you can no longer see it, and then just also check that the condenser aperture is open as well, that gets you the highest NA in the condenser, and that will get you the highest resolution. Now, obviously that is a bit of a very condensed set of steps for lots of different microscopes. They're all different, but essentially these, you can identify these sort of stages on every microscope, and they might not necessarily allow you to adjust them, but you can identify them. And optimizing those will optimize the certainly the white light performance of the microscope, you know, when you're using a lamp, sample, objective, eyepiece type arrangement. What was very early recognized is that, you know, lots of samples don't actually have any contrast to them. You know, they basically, when you put a thin slice of tissue or a thin slice of anything, and effectively it just seems transparent. You know, a typical cell culture monolayer is almost invisible in white light illumination. What was realized, I guess relatively later on, was that you could actually use interference to bring contrast into these samples, and it's based on the idea that your sample, your cell, or whatever it is, has got a slightly different refractive index to the surrounding medium. That's the water, let's say, and because of that refractive index difference, there's a phase retardation, and then that can be exploited to do interference. So that led to these sort of two dominant schemes. One is phase contrast from Zernica, and then differential interference contrast from Smith and Nomarski. So I'm just going to quick, but these are basically interferometers built onto the microscope. So the idea with phase contrast, so this is what you typically see in cell culture. If you look at this sort of picture down here, there's a no phase contrast, you can sort of make out the dead stuff, but in phase contrast, you can get this sort of much more contrasty images, and you can see how the cells are spreading much more reliably. The concept is that you basically put an aperture on the illumination side, on the condenser side. So here it's a sort of ring-like aperture, and then you illuminate the sample with the light from that through the normal fashion with the condenser. And then in your objective, you have a matched ring somewhere in the back focal plane of the objective. Now that ring isn't actually a stop, it isn't a black ring, it's actually a phase plate. So it's a slightly higher refractive index ring, annulus. And what that does is, conceptually, you can see that unscattered light comes from this annulus down here, goes through the sample, and then goes through this annulus and is phase retarded and up to the eyepiece. But in terms of what's going on at the sample, there is actually light scattering from the sample. Some of the light gets diffracted and refracted and what have you. Sort of it follows light paths that are no longer this sort of conical path that I've just described. But when they get back up through the objective and up to the eyepiece, they're all going to interfere because they come, this is conjugate plane to this plane, this is an object, this is its image. So there is this interference at the image plane between those different light paths. And because of this retardation, you get effectively destructive and constructive interference, and that brings in this contrast into the image. So this is a sort of another version of that sort of explanation. We put in a plane wave onto a uniform plane wave onto the sample. The sample has got a different refractive index to the surrounding media, so it retards it a little bit. It goes through this phase plate and is further retarded enough to get you destructive interference, and you can see contrast in the image. So that's typically what goes on cell culture monitoring. Differential interference contrast is a slightly different sort of interferometer. It's known as a sort of shearing interferometer. So conceptually, if we think about a sort of point source illuminating a point onto the sample and going up to a point in the image plane, what the actual differential interference contrast optics do is shear the beam. So you get basically two slightly shifted beams, and that shift is tiny. It's of the order of half the diameter of the point. So something at the diffraction limited level, and they're split by polarization. So you've got sort of one polarization and the other polarization are passing through the sample, slightly shifted from each other, and then they're recombined by a set of sort of reciprocal optics up the top here, and a polarizer is used in an analyzer in this case to in front of the detector. So this idea is given again here. You've got sort of this can be a white light source. First of all, it's filtered into a single polarization, then it goes into a Wollaston prism. And there's a special version of these called a Numerical prism, which sends out that light. It's at 45 degrees to the polarization coming in, which is why when you look at the microscope, you always see that there's sort of at this angle to the front, back, side to side axis of the microscope. The prisms are always sort of at this 45-degree angle. That is sufficient to split the light into two slightly different. There's my mouse gone. To resolve it into two different polarizations, which go through the sample and then are recombined, and there's an analyzer in front of the eyepiece. What happens is if those two sheared beams go through the same thing, effectively the thickness of the cell in this cartoon here, they're seeing the same phase retardation. Likewise, if they're just going through the media, the water around the cell, they get the same phase retardation. But if they happen to go through an object, through the edge, so one beam is sort of going through the edge of the cell and one beam is going through the water, there is a relative phase retardation. So when those two beams recombine, they can recombine, destructively or constructively, and you get effectively increased contrast at the edges of objects. So here is some idea of what goes on in a DIC microscope. Now, if you pay attention at this edge here, you can see if it's set in if it's adjusted one way, you get a bright edge here. But if I adjust it a different way, I get a dark edge here. And this sort of shading across this diagonal gives us this sort of impression of pseudo 3D effect. The prism itself is basically on a slider, so you can fiddle with that beneath the objective to change the contrast in a way that is most pleasing to your eye. So there is no fully correct right or wrong way. It's just adjust it so you can see contrast in the things that you need to see contrast. What is amazing about this technique is it's surprisingly good at optical sectioning. So you can actually you can distinguish between the surface of a cell and the internals of a cell, as illustrated by this set of images through different focal planes. So it's really gets a lot of contrast. Okay. So up to now, we've really just talked about white light microscopy. What about making the thing more interesting, more useful? Can we add contrast to highlight specific molecular species? Can we sort of highlight cell walls? Can we highlight organelles? Can we highlight particular proteins in there? And obviously, I mean, I think everybody really knows that you can do this. And the way you do it is by doing fluorescence microscopy. And normally we basically put in a molecule that has some sort of targeting function, such as antibodies or naturally occurring toxins or binding partners or actually just GFP, for example. And each of those sort of staining molecules has a fluorescent moiety attached to it as well, so that we can light it up. And that way we can get molecular specificity. So the real advantages of this is what's real killer. It's got, you've got good resolution of the microscope. It's high sensitivity and specificity. I mean, you can actually detect single molecules with this sort of method. Very high specificity. I mean, if you've got a GFP tag on a protein, you know with high degree of confidence that that's when you see green light, that's what you're looking at. And furthermore, it's compatible with living things. And this is the big distinction between sort of light microscopy and electron microscopy, is this notion that we can look at living things with the fluorescence microscope. So what we're using are basically fluorescent dyes. They're sort of molecules like this, sort of bunch of benzene rings that, and they've been messed around with and tinkered with. So we're sort of, probably this would be a first generation molecule, something like fluorescein. Alexa 488 was a sort of a modified version, might be second generation version of a green dye, and probably now we're on sort of third generation. And what they're doing is really playing around with the side groups in order to improve their resistance to photo bleaching or change the way they can permeate membranes and things like this. So they are still being adapted. But what they have in common is this sort of notion of an excitation spectrum, the green dotted line, and an emission spectrum here, the light they emit. And you can see that even something like fluorescein is pretty broad emission, but we can excite with a lamp and a filter set somewhere over here and put a filter set somewhere over here to collect the emission. That is sort of described in these sort of energy state diagrams called Jablonski diagrams. So we have the sort of ground state of a molecule is excited up by, say, blue light into the S1 state. It relaxes down to the bottom of that state. And then from there, it can get back to the ground state by emission of a photon. Now, this photon is basically lower energy than the excitation one. So you get this conversion from blue higher energy photons to green lower energy photons because of this band gap. And then because there's structure in the ground state and the upper state, there's a spectrum. This effect can occur sort of from here to anywhere in the ground state and occasionally from somewhere up here. There are other completing relaxation processes. So photo bleaching, for example, is the one we don't want, and that can often be an effect of something called intersystem crossing, where you end up in a triplet state, and then you go out of this triplet state into a state that just never ever fluoresces ever again. There's quenching, and there's other ways of getting back to the ground state. But so obviously, when people are messing around with engineering the molecules, they're trying to make this process the dominant one. That's a sort of the sign of a good fluorescent label. So often in a laser microscope, you just got this single laser line, and then you've got considerable flexibility to put your sort of emission filter in just to get all the green light out of this microscope onto your detector or the eyeball or a camera. And then you can multiplex, so you can have sort of 488 excitation, 561, and 633 excitation. Then in between those lines, a detection band, and you can have 3 colors. And then typically we might get 4 colors out, so there might be sort of Dappy or Herx, something sitting down here. And then nowadays there are more dyes sitting up here in the sort of further into the infrared. So this is stuff that's invisible to the human eye at this point. How the microscope actually works in terms of doing this is you have this sort of typical filter cube stuck in that so-called infinity space that I mentioned earlier on. So you have excitation light coming in from the back of the microscope, reflected down to the sample. The fluorescence light is a different wavelength, so it comes back up through that filter, that reflection filter, and up into the eyepieces. So you have this sort of a little cube with 3 filters in it. An excitation filter at the back here, which separates out, say, blue light for excitation of GFP, the dichroic, which separates the 2 different colors, and then an emission filter, which just sort of blocks out only the green light getting to the eyeball. Those are in these sort of little cubes, and then you have a turret full of these cubes, and they rotate round, often on a motor, so you can see lots of different colors on your sample. And each of those colors is some specific target, and that's the important part. It's a specificity of targeting. All right. The one problem we do have is this notion that, you know, as we get to bigger and bigger things, thicker chunks of sample, you get more scattering and absorption and out of focus fluorescence, and that's going to sort of obscure the detail that you want to see. And this was definitely a problem as you went to sort of thick tissue slices. Single cells, you can see a lot through a regular fluorescence microscope, frankly. So, you know, confocals and what have you are not necessarily needed. But once you start to get into slices, this effect of out of focus fluorescence is really starting to kill the contrast in the sample. So you're losing this sort of notion of being able to identify specific things. Even if you focus down to a single spot, so here's our sort of target focal plane, you focus all the light down to a single spot there, you can see that above and below the spot is all being hit with the excitation light, and that's all generating out of focus fluorescence. In the 50s, there was this chap called Marvin Minsky, who's actually famous in the sort of world of AI, and he wanted to understand how the brain worked, and he was obviously starting to look at the detailed structure of brain tissue, but, you know, he realized that, you know, he just could not see the details he wanted to do, and he wanted to make a better microscope to allow him to understand structure in the hope of understanding function. And, you know, this is the goal that we're still working on 70 odd years later. So the big thing that he contributed to the field of microscopy, which is outside of his contributions to the world of artificial intelligence and what have you, is that he has said, well, why not illuminate just point by point, so you can reduce the size of this this sort of area of out of focus excitation, and then use a pinhole to get rid of the out of plane light. So just let the light that's conjugate to this part here through to your detector. And with those 2 concepts, the confocal microscope as we understand it today had been invented. So here, this is a sort of typical confocal microscope. You've got an object focal plane with a sort of object point, you know, somewhere or another. You've got a light source that illuminates that, and it's focused down to a point. Because you're only doing point-like illumination, you've got to have some sort of scanning mechanism so that you can build up a 2-dimensional picture. And then up on the detector side, this is where the big difference is. You have a pinhole that only lets light from this point through and then onto the detector. So a detector is sort of now not an imaging detector, but it's just a bucket detector. It's so how much light gets through. And then you give it the coordinates from the scanning mechanism, and together you build up an image, 1 point illuminated, 1 point detected. And so this, in the sort of jargon of this thing, these are conjugate planes, but that was reduced to just confocal planes, so hence the word confocal microscope. This is Minsky's actual contraption. Most of it, this great bridge here, is all about the scanning mechanism. The confocal details are actually down here, which you don't easily see, but you can see that from his sort of pattern picture. You've got a point, a light source reduced to a point, which is then imaged onto the sample. This gantry mechanism would do the sample scanning, and then that's imaged onto another pinhole and then a detector. So here's one objective here. The sample is actually would be mounted in this sort of fork that you can just about make out, and then this objective here is the second objective, and the detector would be behind that. And then this gantry is effectively an electromagnetic scanner that scans side to side and up and down. And from that, you can build up an image. But that's definitely, you know, conceptually what a confocal microscope is like. What it gets you is you go from this, where your sample is lost in sort of sea of fluorescence, to this, where you can see the individual cells lighting up. But it's definitely the combined effect of this sort of point-like illumination, scanning, and the pinhole. The pinhole itself is very important. What the pinhole really does get you is this rejection of out of focus light. So if you think about it really, you know, in terms of the focal plane, the brightness of this spot, it falls off as z squared. So this spot diverges as z squared in terms of the actual intensity. So the highest intensity is here. So the highest intensity generation of fluorescence is here. So that's a z squared phenomena. Again, if you think about the pinhole, you've got this z squared effect as you go in and out of focus relative to the pinhole. So in total, you've got this sort of intensity falling off as z to the fourth. So that's what really gives you this strong sectioning idea, the idea that you can get a three-dimensional slice from a single slice out of a three-dimensional object. So here, you know, conceptually, we've got this point here, the image to the point there, and goes through the pinhole onto the detector. An out of focus point here, slightly below, is focused actually below the pinhole and diverges, and most of the light that it generates doesn't actually get through the pinhole. So you get this sectioning effect, and that's a very important thing, because that allows you to take a stack of 2D slices and build them up into a 3D representation of a three-dimensional object. So that's the real power of a confocal. There's a long history of confocal microscopy at the LMB, and this is what the contraption looked like. So their actual microscope is here, that little tube there. This is all the scanning mechanism. There's a rotating mirror scanning here. This is a light source, which is a laser, and the detector is back here. So if you have a look at that as a diagram, you can sort of see this mechanism here. You get a laser beam, comes onto one galvo for doing Y, a rotating polygon to sweep the beam across in X, and then into the microscope itself. But what you can see is that as the fluorescence comes back off these moving mirrors, by the time it gets back to here, it's now stationary. So you just have a stationary pinhole and a stationary detector. So it's mechanically, it's a very easy, it's an easy system to deal with. This is what the sort of first prototype, first commercial version of a sort of MRC derived confocal looked like, manufactured by Biorad. So this in its day was a revolutionary instrument. The ones we have nowadays have the same principle, but just are much more sophisticated, and the computational aspects of them are vastly improved. So this thing had a sort of frame store to store a single image. Now we can take thousands of images without any problem on these instruments. So what it gets you, you know, a single slice out of a 3D object, multiple colors, no out of focus fluorescence, diffraction limited resolution, and so on. They're really very powerful instruments. You take a bunch of those slices and put them together, you get a depth resolve stack, and you know, this is where a lot of information then suddenly becomes apparent. You can see the three-dimensional structure, and that gives you a much better idea of how things actually are working. When using it, what we've got to really think about is this notion of diffraction limited work. So we have a sort of point source in the sample emitting light. The image of the point source isn't another infinitely small point, it's this diffraction pattern, so-called point spread function or Airy pattern. And what we've got to do is match the pinhole to this pattern. So going back to that sort of concept of the confocal microscope, we've got this pinhole here matched to it, but we've got magnification in, so our object is tiny, and then all the magnification of it means that the actual pinhole itself is somewhat larger, and that basically goes on you see the Abbe equation here, and you can see the objective magnification, confocal magnification means the pinhole itself is of the order of several hundred microns in diameter, well, 50 to 100 microns. Conceptually, rather than talking about these actual physical dimensions, we talk about the area units, which is really this diameter here is the pinhole if it's 1, if it exactly matches that, 2 if it's twice as big as that, 3 if it's 3 times as big, or and so on. And the ideal setting is about 1 area, so this whole peak gets through the pinhole and onto the detector. So that sort of notion is given in this slide here. You can see a pattern and a pinhole, and it's matched. And then if you open up the pinhole, you can see that it's not going to get you more resolution. It just lets through more light in terms of it lets through more background light in this instance. So it's about rejecting background. So 1 pinhole, 1 area unit is the ideal setting. And that sort of shows you here. Power through the pinhole is something of the order of 80 something percent at 1 area, and then after that you get a very small improvement. Right, I'm on time. Okay, so I have actually reached now, but I will quickly move on and press on with something. One thing about the confocal is, so you're obviously welcome to leave if you wish to, but one thing about the confocal is you've got this notion of zoom with electronic zoom. So you've got a spot, looks like this in X, Y, that in Z. Your image is just a grid, and you want to try and match the spot and the grid pixelation so that you sensibly scan through your sample. What you don't want, what you want is sort of basically a sort of 50% overlap between the spot, between each measurement of your sample. Because in the pixelated images, they're just pixels, they're all plotted one next to each other. You don't get any sense of this overlap. If it's too small, you're just taking an extra, you don't get any more extra resolution, it just takes longer. If there's a gap, this is particularly bad because you've missed information in these gaps. Again, you don't see that in the pixelated image that you're getting on the screen. So this is a bad system. This is known as undersampling. Fortunately, the microscopes now basically have a sort of button that optimizes the scanning for your particular choice of wavelength and objective setting, and likewise will optimize the pinhole setting for you. There's a whole bunch of sort of points from going from your sort of sample, scanning it with a laser, up to the digitized signal that gets into that is displayed on this computer screen. So this is why you get training. We basically train you how to set all of this stuff correctly so that you get the best representation of the of your sample on the screen. Because really, these are all user variables. The only thing that you don't interfere with is basically the HD converter itself. You can change gain of the detection side and laser power and so forth. So, and the pixelation. So it's important that you get trained in order to use these instruments as usefully as possible. But yeah, if you've got a noisy image, really you've got to put more light in. If photo bleaching is killing you, you've got to put less light in. So those are the sort of two things that right at the beginning of the process. The other thing that we'll do in the training is this idea of getting rid of saturation. You don't want to be clipping the dynamic range of the instrument by having too much gain in your detection or too much laser power, because this just ends up with saturation. So things where if you don't have the bottom level, the offset set correctly, you just clip down here at the bottom. We don't have too much laser light on or too much gain on your detector. It just clips it off flat at the top there. So this is a bad situation because you cannot recover the information about, say, this peak or down here in the feet from a saturated image. And the final topic I'll just go through quickly is this idea of two-photon laser scanning microscopy. This was invented around sort of 1990 by Denk and Strickler and Webb, and it's a different way of doing fluorescence. And what it basically boils down to is if you put a lot of very high energy, a lot of low energy photons that don't have enough energy themselves to get into the excited state, you can go via, if you get them concentrated in space and time, you can effectively get two of them to get you up into the excited state and then get fluorescence emission. And what this means is this notion of concentration in space and time means that effectively you only get excitation at the focus. Away from focus, they're basically too spread out for this effect to have any chance of happening. How you actually achieve that is by using a pulsed laser that gives all the energy of the laser comes out in very short pulses, so typically 100 femtosecond duration pulse, with very, very high peak power. You combine that with focusing and you get excitation at the spot. If we compare this to this one photon excitation with this out of focus fluorescence, you can see that rather than getting a sort of a continuous excitation into focus and out of focus, you just get this point-like focus. So that's the real advantage of two-photon, is depth resolved excitation. But the other thing, because you're using near infrared light, you can effectively beat scattering absorption. So you can actually work in sort of physiologically intact tissue and eventually in living animals. So the big advantage is deeper penetration into scattering samples. And then this is sort of indicated by the sort of idea of water absorption and hemoglobin absorption. That sort of hemoglobin is dropping off as you get into the infrared. Water has got this peak somewhere here, and then between the two is the spectral window where multiphoton microscopes actually work. Nowadays, they'll go out much further into 1500 nanometers. Your excitation way out here, and your detection is back in the visible again. So you can use simpler emission filters and excitation filters. This is one example, looking at atherosclerotic lesions on a bit of aorta, these sort of red stained plaques. You can actually look at aorta directly by autofluorescence. So use two-photon excitation generating autofluorescence, and there's also another effect called second harmonic generation. This is what you see if you do an Z section of it, but I just wanted to illustrate why the sort of three-dimensional properties of two-photon imaging and of confocal in general are important. It comes from this movie. I mean, often it used to bother, how is it that white blood cells can get through the wall of an artery and get into the middle of the artery? And you can see that once you look at this as a three-dimensional structure, that the answer is there in front of you, but it's certainly not apparent from the single slices. I can see that I'm being told to stop. The big advantage of multiphoton is the fact that you can use it in live animals and live model animals. So this sort of indicates sort of where people have done multiphoton light microscopy all over different organs. But the real game changer has been in the brain. For brain research, multiphoton into the exposed brain of freely behaving model animals has been a real game changer, and that's what Denk was trying to do when he was coming up with this invention. So on that thought, I'm going to finish and say, please answer any question. Please give me any questions, and I'll be happy to attempt to answer them. I'm just going to have a look at the. I'll look at the Q&A. Oh, nothing. Chat, nothing. You can hear me. Okay, here we go. Confocal slice thickness is the focal depth of focus from the lens. What would be the focal depth or depth of focus in classical fluorescence microscopy? Okay. So, yeah, you're right. The lens itself, the light comes down into a focal plane and then diverges off, and then your detector is conjugate to that. So in many respects, that focal plane is determined by the lens itself. What the confocal pinhole does is takes away all the out of focus stuff behind and in front of it, and that effectively gives you improvement in contrast in that focal plane. And that's the difference between regular wide field fluorescence microscopy and confocal microscopy. So now if you imagine you've got single cells grown on a cover slip expressed in GFP, you can indeed distinguish different focal planes as you go through them if you're using a high NA objective, something like 100x or a 60x oil immersion or 60x water. You can focus up and down and truly see the difference. If you're using a 10x with this sort of much lower NA, so the sort of much less of a wide angle, this would be a sort of oil immersion, this is a sort of 10x. That focal plane is a much broader thing. You don't see that difference. But where it really comes in is when you've got this combination of a high NA lens with sort of coming to a focus of very steep angles and then going out of focus, plus the rejection of out of focus fluorescence, that really helps you get a better looking image. So it's a lot to do with the rejection of out of focus light. The confocal pinhole does indeed give you a bit more resolution, but I don't really go into why that is, but it's because you have an excitation point spread function convolved with a detection point spread function, and it's the combination of those two that gives you the extra resolution in a confocal microscope, whereas in a classical fluorescence microscope, those functions, the detection, the excitation function and the detection function are basically much more unbounded, and so you see more and your resolution is less. So I hope that is somewhat of an answer to your question. If there are any other questions, please go ahead. No? No more questions. Anyone who wants to stick their hand up, feel free to do as well. Other than that, if there are no more questions, feel free to email me or any of the team, and we'll do our best to answer your inquiries. Otherwise, I will hand back over to Chris.
Okay, thank you, Nick. So we look forward to seeing you again on Thursday at the same time, 10 o'clock. And it will be, oh, right, we have another question about whether or not we have two-photon microscopy at the LMB. We do have two-photon microscopy at the LMB. So it uses a water dipping lens, so most of the time. So this is sort of optimized for looking in at thicker tissue samples. But yeah, we do. So it's an upright microscope dipping into the sample from above. But yeah, we do have that. And then some of the research groups have their own systems, but those are basically dedicated to particular experiments. Ours will do our best to adapt it to any sample you have. Thank you. Okay. Okay, I guess we'll close the meeting and reconvene on Thursday. All right. Thank you very much. Yeah. Okay then. Bye. Bye-bye.